Got it. Today, I would like to speak about our efforts to minimize sample consumption for time-resolved solution scattering. This is, before I start, I would like to acknowledge people who participated in these efforts. It's firstly BioCARS staff from University of Chicago, Robert Henning, Ince Kim, Vukit Astryer, and Guy Meisha. For the sample commissioning for this setup, Sion or Kim from IHE group produced a sample. Pilot experiments for mixing using this cell were done in collaboration with Professor Lin Chen group, Arnold Chen, Adam, and Madeline Ho. We discussed this co-flow cell with people from Australian Synchrotron, Nigel Kirby, Tim Ryan, and Jessica Hopkins from BioCARS. And the cell was produced by Michael Kussel from Kussel Design Australia. So I would like to acknowledge all those people before I start. The outline is pretty simple. I will speak a little bit about our hardware, about typical pump probe X-ray solution scattering methods, about current BioCARS time-resolved solution scattering sample environment and sample consumption. I will describe the co-flow cell and how it was commissioned and how data is comparable or not comparable with time-resolved data, which is collected in using standard methods. So this is a slide which basically modified picture from Tim Graeber BioCARS facility paper. APS deliver at X-ray pulse train, and it takes one single bunch about 3.6 microseconds to traverse around the ring. The power of this X-ray beam delivered by APS is about 500 watts, and if it's focused as it is, this is enough power to melt beamline equipment. And so in the very beginning, after the upgrade, we the high heat low chopper custom design was put together, which basically a disk with different patterns of openings, which allow a burst of 22 microseconds, 1 watt power downstream to our equipment. Then the X-ray custom design ULIC chopper is 1 kilohertz shutter, which is capable of isolating and delivering single X-ray pulse or more on demand. And millisecond shutter is actually fast shutter with opening time of about 4 milliseconds, which is basically data collection shutter. And in this picture, the red figure represents the sample, which can be flow cell or a crystal, which is mounted on the goniometer, and laser beam in this picture is delivered in orthogonal geometry to the beam. We use Rayonic CCD detector, integrating detector to record our scattering patterns. I will speak a little bit about the characteristic timescales, and then I will reuse this picture in regards to how much sample we potentially can consume for different type of the experiments. Here, I would like to emphasize the pump probe time domain, and when I'm talking about pump, I'm talking about any pump. So when rapidly pressure can change, temperature can change, pH can change, and so on. At BioCARS, we normally use laser to initiate all those temperature jump or pH jump, or just changes in chromophore, which then propagates through the molecule, and then we look at the molecule. Here I will speak a little bit how we collect the data. As I discussed earlier, in standard 24 bunch mode, 24 100 picoseconds X-ray pulses evenly distributed over 3.6 microseconds. And the spacing between them, as in this picture, is about 150 nanoseconds. So heat load chopper cuts the 22 microseconds, and a single and ULIC chopper can cut a single 100 picosecond pulse, X-ray pulse, or more. We normally can do, I think, up to 24, 48 X-ray pulse, train of X-ray pulses on demand. The in time resolved solution scattering experiment, the reaction at BioCARS, the reaction is normally initiated by the laser and probe the X-ray pulse, and the time between the laser pulse and X-ray pulse is actually time delay. So the reaction is initiated by the laser, probe X-ray pulse, then the sample is refreshed, and then process is continuous, and repetition for this process, repetition rate, and that can be defined by the users. All the synchronization, complicated synchronization between laser shutter, X-rays, time delays, other equipment is taken care of by field programming gate array. And that was designed and implemented some time ago by Friedrich Schotter. I think he started at ESRF and then moved his expertise here to the BioCARS. The time resolve signal is usually very small, and it takes about 2,000 single pulses repeats to collect one image. And so this basically defines and limits sample consumption. Normally we collect, the software collect multiple time delays in passes, and to collect data for one time delay, we probably collect about 50 passes on average. It really depends on the time resolve signal and the statistics. In each pass, we normally collect off image. It's either negative time delay when the X-ray comes first and laser comes second, or it's just off images. And then software corrects an average images, normalized, then takes on and off, and we have arrays of on and off time resolve scattering difference patterns as a function of time delays, and then process this data. We have several solution scattering setups. The very first solution scattering setup was put in 2000s when we used a capillary which contains the sample, and then we refresh the sample by scanning the capillary through the beam. So in this setup, we see an old cone and cryogel to stabilize the temperature. The laser comes in orthogonal geometry, and this is X-rays, collimator for the X-rays, and this is the laser. Nowadays, obviously the sample has to be not radiation sensitive, and the chromophore shouldn't be damaged by the laser light, because this capillary exposed, I would say, 1,000 times. I think one of the first samples which was collected there was PYP. Right now we are using the standard cell, where capillary basically is replaced by the flow cell. And we either can flow sample while we collect the data, or more standard way of doing measurements is to refresh the volume of the capillary between translations of the capillary. We also have a thermal cell where capillary is attached to the thermal device, and therefore we can change temperature of the sample. Mostly we use that for the temperature jumps for folding experiments and so on. And new setup is a co-flow cell, which I will discuss shortly. And in this flow cell, we do not translate flow cell, we just flow the sample stream in the middle of the buffer stream. And I will discuss this process in details. Before we go any further, I would like to briefly describe fluid dynamics in the capillary, just to justify why we are doing things this way. This is very very standard standard setup when and for the incompressible laminar flow in the capillary or tube, the problem is decided analytically. I will not go through the equations, but I would like to tell that important boundary condition which creates a lot of problems is that the speed of the liquid on the boundaries with the capillary equals to 0. So we can calculate the average speed by dividing the size of the laser spot or whatever we would like to take, because we don't want to expose the sample 2 times, and divided by the repetition rate. So this is the volume which has to be, this volume has to be replenished between the pump probes. And from that we can calculate the velocity profile, which I have here on the top graph. Right, I calculated that for different frequencies which is available at our beamline, from 1 Hertz. Here we can do a slower if we want to 1 kilohertz. And on the figure bottom, I just divided this length of the laser spot, transverse laser spot, by the velocity profile, just to have an idea what happens in the capillary. And we see that outer portion of the capillary is heavily affected. Firstly, the volume, because of the decreased velocity of the liquid in the capillary, this volume is probed multiple times between the pump probes. So therefore, there is a problem with the radiation damage, and also the problem that potentially the sample can experience multiple pump probe cycles, which is also not good and can affect our time resolve signal in at least partially. So if we flow the sample in the middle of the capillary, for example, where there is the dashed line, the outer portion will be the buffer. And if we will look at the top screen, we will see that everything in blue is the velocity of the buffer, and everything is in black is the velocity of the sample. Therefore, we expect that it would mitigate not only radiation damage, but potentially some minor problem with the collecting time resolve signal. I would like to discuss, because of that, we have potentially drag of the sample and do not replenish the sample while we flow continuously. We, in most cases, we choose to do the experiment when we will refresh the sample in the capillary, then go through the pump probe cycles, by translating the capillaries through the laser and X-ray beam, and then refresh sample again. Because we would like to ensure that all sample is refreshed, we pump in large volume that we normally do it, which would normally required and defined by the flow cell. For example, for 0.5 millimeters capillary cell, we each time we refresh 20 microliters, and the cell is about 8 to 10 microliters, and it takes about 100 steps to collect the data. So for the 2000 pump probe cycles, it would require 20 flushes in this 0.5 millimeters capillary cell, which would be 0.4 milliliters. It's a very big value. And so if we will think that we need 50 images to collect time result data, then it would be 20 milliliters for only one data point. In this table, I basically presented a time resolution which we would like to achieve in our data measurement. That depends on our time result process, and what X-ray bunch can be used to match this time resolution, and of course what sample consumption will be for 1 millimeter diameter capillary and 0.5 millimeter diameter capillary. If we will pump appropriately more sample, then it to mitigate this effect, drag effect, and poor sample replacement at the capillary wall. 1 millimeter capillary diameter, it's I don't remember when we last time used that. We normally use 0.5 diameter capillary or 0.7 millimeter capillary. And so to collect one data point for the process which would require more than 3.5 microseconds time resolution, we need 0.8 milliliters per time delay, or 20 milliliters per time delay if we cannot reuse the sample. And therefore, that's very important. Right now, we can work only mostly with the sample environment and samples which are reversible. Because if the sample is reversible, the sample quantity is significantly reduced. I think the minimal quantity which we used at our beamline was about 300 microliters to collect data in the standard pump probe typical experiment. Yes, and I use this time K6 time scale picture just to show that in this time domain where we have basically photosynthesis, electron transfer, signal transduction, I think motion of helices or beta sheets and so on, small protein. So it would require, basically, if we cannot reuse the sample, it would require 250 milliliters of sample. It's just absolutely not possible to produce if sample is not commercial. And of course, this is minimized 24 times when we're studying processes which are more slow and where we can use more bunches at once. Again, for slower signal transduction and for slow protein folding, ligand binding, and so on. Here, I would like to introduce the co-flow cell, and in co-flow cell, the central part of the cell is occupied by the sample, and outer port of the sample is occupied by the stream. I would like basically the sample solute is injected through a tube or a needle into capillary filled with solvent. So this is using CoFlow type cell. Of course, it's absolutely not a new idea. It's used for a very long time for tailored dispersion analysis, cytometry, self-sorting, fluorescence, flow-induced dispersion analysis, many other different applications, including commercial applications here. In X-ray solution scattering, CoFlow device, which is used for static in static sacks, was used by scientists from Australian synchrotron, Nigel Kirby and Tim Ryan, the names, I think their cell also is used by Jesse Hopkins at BioCARS, and I discussed with them this cell before we proceeded and designed and utilized our cell. So we worked with Australian company Crystal Design, and they constructed, designed this Sheath flow cell in the way as it is. So this cell can be mounted either horizontally or vertically at our beamline. It can intercept pseudocollinear, collinear, and orthogonal beams. It is compatible with our visualization optics, helium cone, and there are a few pictures here how it is mounted horizontally or vertically, and it consists basically a support which can be mounted one way or another. The sample is injected through the HPLC needle 90 degrees. There is a sample buffer line which is going in this volume. There is vent. The capillary is mounted to the support, this sturdy support, and leak proof with the O-rings, which worked well so far. And there is just basically discard line. Initially, we commissioned it with the sunset yellow dye, just to see how it behaves. And one of the pictures is basically here. This is another slide with the same view. This is flow cell mounted in provided support, and there are a few images from Crystal Design. This basically shows the guts of the flow cell, how the capillary is mounted inside the structure using O-rings. The capillary is 600 microns in diameter, and the setup so far was very robust. I will present some calculations for the CoFlow experiment sample consumption. Right here, we just calculate how much sample will be flown in the similar geometry. Normally, we design this cell to accept 300 micrometers, so 0.3 millimeters diameter sample. And just pure calculation shows us at least 10 times a decrease in the sample consumption. For example, for the single bunch measurements, instead of 20 milliliters per time delay point, we have 1.75, which is still, of course, big value, but much more achievable than standard flow cell value if the reaction is irreversible. And the pH jump, or for example, cage compound ligand binding, enzymatic reaction of some sort, all these reactions which are very interesting to study, they were often irreversible, and we have big problems studying those with a single X-ray bunch. Going further, if I will go back to characteristic time scale, so instead 250 milliliters for the data collection from 12 data points, we can see that it is like 15 milliliters perhaps, and also below 1 milliliters for 24 bunch mode. We used photoactive yellow protein solution for this experiment to commission the flow cell, and I said that photoactive yellow protein solution was produced by Seon Kim from Professor Ahe group, KAIST, South Korea. In the standard geometry, we used laser illumination from above, and this is colorimeter with X-rays. This is the standard flow cell, which is scanned across the beam. We use nanosecond laser, 7 nanosecond pulse length, 400 nanometers power, I think 1 millijoule per millimeter squared, and standard laser beam 120 by 500 micrometers, where 500 micrometers is along the beam and 120 micrometers is across the capillary and perpendicular to the X-ray beam direction. Flow cell was translated between pump probes, and sample refreshment was 50 microliters for the 0.5 millimeter cell. The repetition rates was 5 Hertz for the longer time delays and 20 Hertz for the single bunch measurements. This is a standard co-flow cell setup where there is a co-flow. The laser was used in the collinear geometry, and X-rays, like we cannot modify the X-rays direction. So it's about 15 degrees between the laser and X-rays. The laser beam was 120 by 120 micrometers. And it was delivered through the fiber, and then we used the focusing optics to focus it at the sample position. So sample stream diameter was 0.3 millimeters in 0.6 millimeters capillary, and sample flow rate was chosen to refresh the sample, and the buffer then just matches the sample velocity to produce the stream of desired diameter, and the repetition rate was basically the same, 5 Hertz and 20 Hertz. For the continuous flow of the sample, to ensure the proper overlap, the software was modified to offset the laser. For the longer time delays, I think for more than 1 milliseconds, we need to offset the laser to ensure proper overlap. And this is the data which I've got. We used 1.5 millimolar PFIP solution and the buffer, 20 millimolar tris, pH 7.0, I think a little bit of sodium chloride. And in the co-flow, we have to use matching buffer. I have to mention that I have to degas thoroughly the sample and the buffer before using, otherwise the flow of the sample is not stable and can be affected potential bubbles. The data collection strategy is basically the same. One image is equals of 2000 nominate as single pulses. That means that if it is single bunch, we collect 2000 pump probes. If this 24 bunch mode, we collect 2000 divided by 24 bunches. 50 images at each time delay, and at 20 Hertz minus 5 minus 10, 500 nanoseconds, 50 nanoseconds, 500 nanoseconds, and 500 microseconds, and at 5 Hertz, like also a string of time delays up to 150 milliseconds. And again, this sample was studied in crystallographic form and in solution form by two our research groups. One group is Phil and Anfenrun group, and another group is Heritek Ike group. And these data were published. So this is a very good sample for just for collecting the data and just to see how it is compared to the previous data and so on. I collected, as you see, I collected only a limited number of time delays, much less than it normally would require to study the whole signal transaction in PYP, but the reason for this experiment was just to compare the data and see how much sample I will get. Sample consumption will be in the two different scenarios. On the left, I shown time resolved solution scattering signal as function of scattering vector for different time delays for the standard cell, and on the right for the co-flow cell. So these data were collected more or less identically, except for the standard flow cell. I collected only 1,000 images per 1,000 pump probes per image. And that's basically because these data collection takes very long time, because we need to translate flow cell slowly to avoid the vibrations, and I was just running out of time. So as we see if we will look at the signal, we will see that it's more or less similar for the same time delay. At 50 milliseconds and 5 milliseconds and 50 milliseconds, we see the biggest signal dip, which is caused by elongation of the protein, so-called signal state, which is can be observed only in the solution scattering, but not in the crystallographic form. So what is interesting here for us is to look at the sample consumption for these two data sets. For the standard cell, for these time delays and identical data collection, I consume 7 milliliters of sample. In the co-flow cell, it's 660 microliters. If I will go to the single bunch, I used 8.8 milliliters of sample and 50 milliliters of sample for the standard cell. And I would like to say that this is very big differences, and I think this cell can be performed very well, robustly, and can be used by other research groups to do their measurements. If they're doing measurements in the sample which undergo irreversible reaction, because once the sample pass through the flow cell, it is diluted. So if you need to recover the sample, the only way to recover the sample is to reconcentrate it. Otherwise, the sample concentration will be different on the output of this flow cell. Data analysis is actually very brief, because there is not enough data to actually do any particular data analysis. And I use the RegLab kinetic program, which is available at BioCARS, to extract intermediates. I use the serial model A to B to C to D. SVD analysis showed 3 components, and basically that's how I model the data. This is not very serious modeling at all. I used the data up to 1 reversed angstrom scattering vector to analyze them. I didn't do any heating signal subtraction and so on. I just got the data at 1 reverse angstrom and use it as it is. Jan-san probably will excuse me that I reused some of the pictures from his PYP work. This is just for the representation purposes. We see the first and second vector, very small changes in the scattering pattern. That means that the changes in the shape are small. I didn't do any reconstruction at all. But I put in this model, I put a co-flow like first state, second state, third state, and then it goes to the ground state. kinetic time constant for the co-flow, I put inside this circle 0.07 milliseconds to 0.2 milliseconds, 0.26 seconds to go to the ground state from the signaling state. And outside is the standard flow cell. And though the data are different a little bit, but really not that significantly. With that, it will be my conclusion. So co-flow cell showed itself as a robust device, which can be used reliably for time-resolved pump probe solution scattering experiments, especially for the radiation-sensitive, viscous, irreversible reaction, which is difficult to do in the standard co-flow cell. The sample consumption in co-flow cell experiments, it's 8 to 10 times less than, I wrote more, less than in standard flow cell. Co-flow cell eliminates radiation damage, potentially can improve observation of time-resolved signal. And with that, I would like once more thank you all the people who participated in the commissioning. And if you have any questions, I will be happy to answer those. Thank you.
Thanks, Irina. Are there any questions? Well, I guess I have a question. Was the PYP data collected in the horizontal or the vertical orientation for this?
Yeah, I collected this in the vertical orientation, and
Okay.
I think the density, I forgot what the density was, 1.5 millimolar. Probably this is closer to 20 milligram per milliliter. Okay. This is much smaller than, for example, was used by Janssen in his PYP experiments. They have to correct the data for the protein-protein interaction. In the horizontal geometry, when we are using more dense sample, I do not observe a good laminar flow. Only low density sample can be used in the horizontal direction because of mixing and fluid dynamics. Okay. So at in the horizontal orientation, we can go maybe 5, mostly 10 milligram per milliliters. I did experiments with dye for these pilot experiments, and therefore for the PYP we use the vertical orientation when the gravity is in line with the flow, and in this orientation it works very well and quite stable.
Okay, great. Any questions?
Hi, this is Friedrich.
Yep.
Yeah, very nice presentation, Irina. Thank you very much. You made some remark about whether it's possible to recover the sample. If I understand right, I mean, the outer flow is just water, isn't it? So it mixes with the PYP solution. Is that correct?
Not exactly water. We are matching the outer buffer exactly with the sample solution. Therefore, because we do not want to change the pH and salt concentration of the inner stream, and therefore, after the sample is measured, if the sample is reversible, it can be put through the concentrator and reconcentrated back. Therefore, I concentrated it for other commissioning tasks and measurements and store it. Yeah. But it's very important to match the outer shield exactly, right? Because otherwise, part of your sample might have a gradient in like salt concentration or pH, proton concentration, which we would like to avoid, because that can affect your time resolved signal alternatively.
Yeah, I was just wondering, why don't you use an immiscible liquid for the outer, like a fluorocarbon that's the same density as water and same viscosity, and then phase separates later?
You know, Frederick, you're right. But you know, firstly, I do not know what will be the chemistry on the boundaries, and secondly, I'm not sure. Like last time I've checked, it was some problems with the like toxicity and flammability of those liquids, but maybe I have to look more closely into this. This is interesting suggestion, actually. Yeah.
Yeah, I mean, there's fluor inert, isn't it? It's completely non-flammable, completely inert, non-toxic.
Okay.
It just evaporates like water. Okay, there's no need to recover it.
This is actually great. So when you don't need to recover your sample, that might be interesting solution. I will talk to you later, Friedrich, just to see what you know, the properties of this organic solvent.
Okay. Maybe you have another question.
Mm-hmm.
When you look at these long time delays, like 15 milliseconds, 100 milliseconds, do you need to scan your laser beam to offset the displacement of the pump volume?
That's what I did. You know, I didn't actually scan it. I just, on the basis of the velocity, which is at this point already fairly uniform, I offset the laser beam, right? And that's how I recovered the signal. And because the laser beam was much larger than the X-ray beam. X-ray beam is standard X-ray beam, like 35 micrometers full width half max, and laser is 120. I obviously didn't have much problems with the overlap, as the data shows. Yeah.
Oh, okay, very good, very good.
Mm-hmm. But yes, I need to offset laser beam. If it would be collinear geometry, that would be vertical position of the laser. I place it on the motorized stage and basically interlocked it in the software. If it's in a horizontal direction, the laser stages will work well for that. Yeah, I don't know what you're doing in your experiments. If you do scan the cell or flow cell, or you flow continuously for your time resolved solution scattering experiments.
Oh, both, isn't it? So, I mean,
both.
For
Yeah.
the short term, we scan. Okay, for the long term, we pump.
Mm-hmm. Yeah. Yeah. Okay.
Any other questions?
Actually, I have one. This is Cynthia Stoffaker, Irina. Lovely
Yes.
talk. Thank you. I have a question. This is set up very well for laser, pump probe sort of things like that. What about, is there any future in the CoFlow cell for things like pH jump? I understand the problem with mixing and the rest of that, but what do you think?
Yeah. Okay. So for the pH jump, we were actually talking about at our beamline pH jump measurements. Published pH jump measurement so far was done by Lin-Chen group. And the pH jump basically was initiated by the laser. The caged protons were used, and then we used UV light to basically release the protons. And in this sense, then it works like a standard pump probe experiment. If you're asking about the mixing, this theoretically it can be done, right? Diffusion and mixing. So in the sense, this is a laminar cell, right? And this is the cell which similar to custom build. This cell is basically commercially produced. If I need to change it, I just replace the capillary, right? Or if I need to modify the capillary and then reuse the everything else. So that's what we like about this cell. But for the mixing experiment, it is not clear, right? So because just because of the diffusion coefficients, right? And so even if we use a proton diffusion for the pH jump, right, the diffusion is 7,000 micrometers per second, right? And I just basically, we measure in the sample, we can do the measurements right now, 5 millimeters from the tip. And then that will be, if I would like to achieve time resolution, for example, 1 milliseconds and less and more, proper diffusion length has to be assessed because some mixing has to occur. And the fusion length I calculated for the protons for different time, and with our beam compatible only maybe 50-100 millisecond time delays, right? And everything smaller than that, we will have a very narrow stream within very wide buffer background, which is basically difficult to measure. And here I was trying to calculate for these streams what has to be the buffer flow rate. And buffer flow rate in this last column, and you can see this red and yellow. This is where basically even vendor even cannot guarantee that the flow cell will not be damaged, right? So our safe region will be somewhere around maybe 50 and more milliseconds. And we did these measurements. We did cytochrome C coflow experiments, pilot experiment, actually quite ad hoc experiment with Lynchian group, where we used the cytochrome C injected through the needle, and we used the buffer. I think that, what is this? Yeah, this is the parameters, how we did it. We did pH jump from 4 and outer buffer shield was 8, and we were looking at the mixing of that. And this measured the scattering signal, and in this sense that is like standard laminar flow experiment where we have to scan along the capillary to see how the signal behaves. So we saw some pH jump associated with the position in the capillary, and for this particular experiment it was I think 100 millisecond time delay. That's what we were able to do. And I don't know, like you would think that potentially if you do not use cage materials, we can like put some inserts like Louis Pollock right now is developing, which would mix buffer and the sample quickly. But I don't remember last time when Kara was talking about that. I think that they also observed like time delay with these mixers on the order of magnitude milliseconds in the laminar cell, if I remember that correctly, tens of milliseconds. So the answer is yes, we can mix with the cell. However, it will be long, pretty large time resolution, maybe 50 milliseconds and more. Yeah.
Okay, thanks.
Yeah, does it answer your question? I mean,
Yes, it does answer my question. Yeah,
yes, yes.
you know, we have a reaction we can slow to be okay at 50 milliseconds.
Yes.
I think.
Yeah, yeah.
But the mixing was a problem. So anyway, I'll talk to you some other time about that too. Thanks.
Okay. Thank you, Cynthia.
Any other questions for Irina? Okay. Well, thanks again for Irina for talking about the latest and greatest on the beamline for solution scattering. And thank you for doing this BioCARS Zoom seminar for us. That's been a very productive seminar series to discuss the different topics that we have at BioCARS. So
yes, yeah, very nice way to exchange information between our community. SAC.
And if
So-
anyone here is interested in talking about their work, let Irina know and we'll get you on the schedule.
All right, and I would like to say that we are talking not only about time resolved work which is done at our beamline, it can be XFAL beam work, it can be SRF material beam work. So if you are interested, we will be most happy to have your presentation here to see what is going on at ESRF at ID9. So thank you,
All right,
everybody.
with that, thank you, everyone, and I'll see you next time.